what we think about the various medical illness, whether it is infectious disease, autoimmune disease, or even neurological illnesses, or psychiatric illness, oncology. So all those diseases have a little bit of in relationship with the microbiome, and evidences are emerging, and so I'll be touching upon those finer aspects. I think I'll get approximately 45 minutes. Okay, then it's fine. And in between, what are the future prospects? Like, how can we improve upon the various aspects? See, we are facing with some problem, but what is the solution? So with this introduction, I'll be going to the slides, and we have, or we are aware of the fact that humans have approximately 38 trillion cells in our body. 38 trillion cells. But we are composed of, or we have more than hundreds trillion cells contributed by microbes. Just think about it at point. And the number of microbes what we have in our body is more than the number of cells what we have. Okay, so in that, with that introduction, like what do you mean by microbiota? Microbiota just means that it's a collection of microorganisms. It includes bacteria, viruses, fungi, archaea, a lot of things. And when it comes to the biome, it means that the genetic factors of those organisms are taken into consideration. So it's the genetics, and the biota means is the organisms. And dysbiosis means that a dysregulated or abnormal microbiota. So as I mentioned, a lot of illnesses it can produce. And what do you mean by this holobiont? Holobiont is something like all the human cells, all the bacteria, all the microbes, and all the fungi, viruses, and all. So all that formulate the term holobiont. And when we look into it, the human cells and the different microorganisms are interacting or is moving synchronously. That means when we have a circadian rhythm, the microorganisms what is inside us, whether it is in the gut or in the skin, ear, mouth, nose, wherever, that also has got a circadian rhythm. So that is something, this sentence is from 21st edition of Harrison, Principles of Internal Medicine. So the pa-di-do, that means synchrony. That is in French. That's the direct sentence from Harrison textbook. And coming to the importance, 70% of the immune response of our body is determined by the gut microbiome or in the gut. And when we talk about various hormones or even peptides or even the serotonin, around 70 to 80% of the serotonin in the body is produced by the gut bacteria. And we all are aware of the fact about an antibiotic associated problems like pseudomembranous colitis. Now we don't say that name pseudomembranous colitis, but we say like Clostridium difficile infection. But once it had happened, or when someone developed that, then to get a recovery from it will take a little bit of time, maybe 2 weeks of more specific antibiotics and time, and even the mortality rate is high. But with the fecal microbiota transplantation, the recovery happens 100% within 48 hours. So it's really a magic to see. So we can offer a solution to the problem like this. And we heard of long chain fatty acids, short chain fatty acids, and medium chain fatty acids. But when it comes to short chain fatty acids, that is entirely synthesized by the gut microbes in the gut. And also, when we look into the gut-brain axis, as Madam has presented, like it's a gut feeling that something wrong in that previous case. So like that, the gut-brain axis. See, when we studied anatomy or even physiology, we thought that vagus nerve descends down, it supplies throughout the GI system. It is a one-way connection in which from the brain to the gut, but it's not so. now evidences are suggestive of the mechanisms in which the gut through the vagus nerve communicates with the brain. So it's vice versa. It's not just one way. It's not only afferent, it's afferent also. So I'll be discussing about the gut-brain axis also a little bit later. So human microbiota, the supra-organism, and I mentioned about the number of human cells, the number of bacterial cells we have, and also about the genes. Now a point about the genes, because it's about microbiome. The genes, number of genes we have is around 21,000. So, but the number of microbes is 100 trillion, and they have around 3 to 4 millions of genes. So we have inside us 4 millions of genes, and we human beings have only 21 to 22,000. So why this disparity? And why this is? Because there is the concept of co-evolvement. Co-evolve means that before human beings there was bacteria, and this bacteria has entered us in a later state, but it evolved along with us, and we have given a lot of our job, lot of our physiological process to the bacteria inside us to do this job. So the number of cells are less, the number of genes which are doing our physiological process is less. We are outsourcing our physiological process to the microbiome. And so there is an involvement of the toxins and how the beneficial metabolites, the immunity and nutrition, and the environment factors all can influence the microbiota in the health status and in the disease status. If we are 10% human, then who really parasites who? So with that, when the discussion is about some science, the co-evolvement, then let it be 2 slides of spirituality also, because when we think about the co-evolvement, there is a little bit of dependent origination and dependent arising. That means the one contains the many and the many contains the one. Without the one, there cannot be the many, and without the many, there cannot be the one. This looks a little bit complicated, but it's very simple. You just think that there is a dependent origination and dependent arising, and this is the fundamental of Buddhist physiology. So also, when we discuss and think more in line, then we human beings are not 100% human being. We have to agree. The evidence is that. So when you go into this, you can see in Ishavyasya Upanishad verse 6, this is Yasya sarveshu budeshu sakshatmanam tado munim sarva budeshu chatmanam tado na vijugupsadare. That means they live in wisdom who see themselves in all and all in them. This also means there is a little bit of interdependence. It's not only the gut microbiome and the human cells and the genetic interaction, but no, this means it's an interaction between us and the environment, the interdependence between the human beings, all the organisms, all the animals, all the plants, everything, and even the universe. Okay. So now coming to the history of the microbiology a little bit, because this was Anthony van Leeuwenhoek who invented or thought of the microbiota. He found out that there is a difference between fecal and oral oral bacteria, and in that itself, the alcoholism and tobacco use, there is a difference between the microbes, and the microbes differ in young and old men. And Louis Pasteur, he found out in 1885 that when a living being is deprived of common microbes, they are unable to live. So this was founded by Louis Pasteur. So from where we go, where we acquire our gut microbiota, this is even before we are born. There is a theory that we assimilate it from the amniotic fluid, and the significance of mode of delivery is very, very important. And there are more and more evidences to show that kids born out of normal vaginal delivery have more diverse gut bacteria, and even during the process of birth, the genital microbiome changes according to each trimesters, and it prepares itself so that when during the process of delivery, the newborn consumes the secretions, and they in turn will have a diverse microbiome, and that helps in taking out of oligosaccharides which are present in the milk, because milk's oligosaccharides are why it's there. It's not easily digestible. Maybe pediatricians can say about it, because it's not easily digestible. It's not just glucose, it's not fat, but it contains oligosaccharides, and that these microbes helps in digestion of. So significance of the mode of delivery, then exposures of microbes during infancy, the theory of hygiene hypothesis, as more and more allergy, atopic dermatitis, asthma, allergic asthma, all those things are affecting the common life or the modern life. The idea of hygiene hypothesis, in which when a kid is not exposed to the dirt of or even the surroundings, then adequate antibodies are not developed, and for that a crucial role is that of the gut microbiome. And so there is a lot of factors which are responsible for the gut microbiome acquisition. And during the human lifespan, you can see that the diversity and richness, it's not the number that matters. So even though we mentioned about a huge number, like 100 trillion, and the diversity and richness is what really matters, because how diverse the microbiome is, or microbiota is, rather than the number. No discussion about gut microbiome is complete without Human Genome Project. It's like it ran from 1990 to 2003. The project, it given human genome identity, and humans have about 20,000 to 23,000 of gene, and which is less than many other organisms like rice and water fleas. So human beings' number of genes are less. Then Human Microbiome Project is like, with the advent of newer technologies and with artificial intelligence, a lot of studies where the different composition of the microbiota differ by different body sites. That is, different body sites have different bacterial populations, and there is tremendously inter-individual variation, and no microbial species are ubiquitous in all individuals. But the genome or the genetics of two individuals are 99.1% same. But the microbial presence, even if one individual is from a group of people are from a single home, it differs. And personalization of the human microbiome, and there is an overall diversity of more than 10,000 different bacterial species, and intestine alone contains around 1,000 species. And like the point which I mentioned, that we have only 20,000 to 23,000 of genes, and there are 4 million genes inside us. And what are the different scientific tools and models to investigate the microbiome? And when we study about the RNA, it's a metatranscriptomics. When we study about the proteins and what the microbes are doing with it, there is metaproteinomics, and a lot of factors into it. But to make things clear, I will point out what is prebiotic. I'll ask you a question. What is prebiotic? Madam, anything will do. What is prebiotic? Say something. Okay, what is probiotic? Okay, not to complicate the issues, see, the prebiotic is something which is inside our food, within our food, which is not digestible. That means fiber. Okay. So this will be taken up by the bacteria in the gut. So what is available for the bacteria to eat in your food is actually prebiotic. Then what is probiotic? Probiotics is nothing, the bacteria. The bacteria inside the gut is the probiotic. Then there is also one more term called postbiotic. That is something important. Postbiotics is what the probiotic produces, or something like the probiotics consumes the prebiotics and form the postbiotics. So postbiotics means it synthesizes a lot of chemicals. So when we discuss about the leaky gut, there are a lot of things into it. The postbiotics, basically the short chain fatty acids, a lot of hormones which we discuss, a lot of chemicals, mucin. See, so the intestinal line epithelium is covered by a layer of mucous membrane, mucus, that is being synthesized by this. So these are the postbiotics. So what can influence the microbiota? Genetics. Our own genetics have a very minimal impact on the microbiota, as well as the age of the age. Younger, older, it has got a. Then diet. Diet is very, very important. But diet, because if you are on a vegetarian diet, certain bacteria flourish in the gut. If you are on non-vegetarian, then another set of bacteria flourishes in the gut. And so what we eat to promote the gut bacteria, depending upon that, certain things. But very commonly, when the food which is available is devoid of fiber, probably it's not good, because most of the fiber related activity happens in the second part, in the large intestine. So when we eat refined processed food, then the amount of fiber which reaches the gut is tremendously less. So good bacteria probably dies of hunger. And also there is another concept that we have some food items which are devoid of calories or less calorie items, or even like artificial sweetness. These artificial sweetness, they give sweetness, but they are damaging to the gut. They are damaging to the gut. So we are, in order to avoid calories, we are switching over to artificial sweetness, but they in turn can damage the gut, and how that damaging can lead to diabetes, I will explain very, very shortly. And certain drugs, see, they're like there are a lot of drugs which changes the chemical milieu of the intestine, and like even statins can change. That doesn't mean we all should stop using statin, but there are evidence that it changes. And also a lot of neoplastic, anti-cancerous drugs. It changes drastically the of course antibiotics. Certain antibiotics, like even ciprofloxacin, when we give it for a day, 5 days, there is a drastic reduction in the gut microbiome variability for around 30 to 40 days. And not only that, what is it supposed, because the action of ciprofloxacin is not against anaerobes. It's not against anaerobes. But the anaerobic bacteria are also affected to some extent. So the effect is too much, and different out of the different bacteria, it affects said to be affecting certain keystone bacterial species in the gut. And when you say about the lifestyle, when we move from India to abroad, say UK, then your gut microbiome changes. When you come back after 3 to 6 months, it's get readjusted to that. And so the lifestyle is very, very important, and also about the stress, then the sleep. So a lot of factors, how it directly influences the gut bacteria, I will be explaining. Then the circadian rhythm, I mentioned about if you have a jet lag, or say like if you're not sleeping properly, or if you're having food at an abnormal time, that can impact the circadian rhythm of the gut microbiome, thereby leading to thereby chemically as well as neurologically or neuroendocrinologically leading to producing some leaky gut, or there's causing some pores in the epithelium so that the bacterial toxins can enter. And the gut-brain axis. What are the different factors affecting the gut-brain axis? Is the food intake, then the genetics and epigenetics and diet, a lot of factors which I discussed before, and the social interaction, stress, happiness, and all those. See, the primarily the gut-brain axis. When you are stressed, why everybody feel like having an abdominal cramp? By hearing some sad news, some news, or he failed in exam. Why that all in the gut? I hope everyone has experienced it. Like she also experienced the gut feeling at 2 a.m. Why? So it's not just it's not the one-way communication; it's the other-way communication. So a lot of factors in it. But when we talk about the gut microbiome, I'll say it's small discussion about epigenetics also, because that is very important. The genetics is what is there destined in our DNA. Inflammatory bowel disease, only 10 to 20% manifest. Remaining 80% with the genes in them, with the DNA in them, are not manifesting. Like that celiac disease. So a lot of diseases with the abnormal gene is not manifesting. But those without the gene is manifesting. So when we look into the genetic code, even in the presence or absence of it, there are a lot of other factors above the genes which can control the genetic expression. See, the genes are there, so there are some factors which can control the expression of the gene, but not affecting the gene. That is epigenetics. So in that epigenetics, it comes a lot of our activities, how we live our life, how we sleep, stress, all factors. And when we look into the molecular mechanism of epigenetics, I think a lot of methylation, acetylation, a lot of things are coming. But keep in mind that our surroundings, our situations, it have a major role in expressing our genes. Next, I will be, just before entering the leaky gut syndrome, I will be saying what is in metabolic endotoxemia. See, the theory almost, when we look into the cardiovascular system, developing metabolic illnesses, the theory goes always in favor of having high cholesterol, oxidized LDL, small dense LDL, producing atherosclerosis, plaque rupture, and all. So, but there is, see, as I mentioned, the gut lining, we have trillions of bacteria, and when the bacterial cell which contain the lipopolysaccharide enters through the pores created and enter the circulation or enter the submucosa, the submucosal lymphoid tissue is activated, and that submucosal lymphoid tissue activation can in turn activate a lot of reactions, including raising the interleukin 6, human necrophysis factor, a lot of things. And this is in turn called as, so in the blood there is an elevation of toxins inside the blood, but actually what happens is in the gut. So this is what is called as the metabolic endotoxemia, leading to dysregulated systemic inflammation and macrophage activation. So basically the immune dysregulation happens. When it's COVID, why some people develop COVID, some other people are not developing COVID. Someone exposed to COVID very frequently, no COVID. Someone who had taken all the 3-4 shots, still COVID, means they develop COVID. Some have very aggressive COVID, some have less aggressive COVID. So there is a theory. So the theory goes in the way like, with the diverse gut microbiome, it secretes a lot of metabolites, the postbiotic. These postbiotics can regulate the expression of T-reg cells. We have the lymphoid tissue, we have helper cells, regulatory cells. This regulatory cells activation is suppressed by the metabolites, by the bacteria. So more diverse bacteria you have in your gut, this activation is suppressed. So the cytokine storm, which you all have seen it during COVID, that is not there. All disease begins in the gut. This was said by Hippocrates 2500 BC. 2500 years back. That means 500 BC. But we people, we start to think about what really made me think about this gut microbiome is hepatic encephalopathy. Titisar is here. So the hepatic encephalopathy means you give a good bowel wash, you wash them, you give them a little bit of lactitol, you give them enema, make them move their bowels at least 3 to 4 times a day, patient improves. So at that point of time, I couldn't find out what really is happening, but we treat the patient like that. So all disease begins in the gut was said by Hippocrates 2500 years back, and now the science is proving that he is correct. With what evidence he guessed it, we don't know, but Hippocrates is proving to be right in 21st century. So a lot of factors and lot of illnesses like neurodegenerative illness, multiple sclerosis, Parkinsonism, Alzheimer's, Huntington's, and metabolic infections, cardiovascular cancers. Okay, and when we look into the modern life, about the genes, uterine environment, life experiences, choices, habits, then a lot of work-related triggers, urbanicity, poor nutrition, a lack of sleep, toxins, emotional stress, hormonal imbalance, infection, a lot of factors. Ultimately, leading to a lot of medical problems. So, multiple line of studies have shown that the primary cause of this environmental associated inflammation may be the dysfunction of gut-brain axis secondary to alteration of gut microbiome. And this is just the same point. A lot of factors influences dysbiosis, leading to a lot of medical problems. And the leaky gut hypothesis. This is basically the first part. The A is the normal intestinal epithelium, the submucosal compartment, the lymphocytes sitting idly, the sensory neuron and the mast cells, and you can see the tight tensions maintained in the integrity. And when it is the leaky gut, you think that the cells have some space, the epithelial cells have some space, so that what is inside the gut can go in the submucosal phase. So what happens is, when it reaches, always think about the lymphoid tissue. 70% of the lymphoid tissue, what the body reacts, is not in the lymph nodes, it's not in the spleen, it's in the gut. So the beauty is trillions of bacteria on one side, just a single layer of epithelium, and downwards you have the strong soldier waiting, the lymphoid tissue waiting. So something enters down to the submucosa, it activates, and the immune cascade, where the mast cell produces cytokines, chemokines. So what it does is the mast cell activation and activate the immune system. And interestingly, you can see the enterosensory neurons there is also activated, thereby activating the enteric nervous system. Also, the metabolites which are produced there can reach the brain through the blood, or can ascend up through the nervous system and reach the brain. So basically, the bacteria, their products, their components entering to the submucosal layer is actually the leaky gut. It's the leaky gut. But it activates a lot of inflammatory cascades, leading to different types, depending upon what part of the bacteria or what metabolites What postbiotic enters, the different diseases manifest. And when it comes to inflammatory bowel disease, as I mentioned before, so the presence of gene is not 100% sure that the disease will manifest. So what really happens is, I mentioned about the T regulatory cells, and normally there is a cytokine production by regulatory T cells and suppresses pro-inflammatory responses. So here what happens is the suppression done by the bacterial variation is lost. So more and more activation happens in the case of IBD. So many of the relevant genes are related to host microbe interactions. And now I think 5 more minutes on. Okay. So obesity and metabolic syndrome, again, in a nutshell, if I'm saying that people who have obesity, when their microbiome or microbiota is transplanted to someone who is lean, then in turn they will have obesity, even if they are taking a less calorie diet. So that doesn't mean throw away your diet. No. Please follow your diet. But this line of thinking is coming up, and that is one point. And so the recent research has implicated the gut microbiota as a critical determinant of nutrient uptake, energy regulation, and ultimately weight and metabolic disorders. So this is just the explanation given to this. Notobiotic mice means mice which are devoid of any bacteria in their gut. So for them, if you transplant the obesity microbiome, they become obese. And microbiota and diabetic disease, this is also, the picture is suggested, just you can see that you can see some, what is a donut and cake there. And that side is the abnormal side, that is dysbiosis. It means that maybe this sort of food can also cause dysbiosis. Because of dysbiosis, a little bit short chain fatty acids which are supposed to be there in the normal side, like butyrate, propanate, and acetate, in their absence, what happens is the LPS, that is lipopolysaccharides, which can enter the submucosal layer, causes inflammation, dendritic cells. So in the liver, it can cause steatosis. In the adipocytes, it can cause inflammation and insulin resistance. So we say that depression is an inflammatory condition. We say that fatty liver is an inflammatory condition. We say that cardiac or a CAD, coronary artery disease, has got an underlying inflammation. So whatever be the condition, then why are we doing high sensitive CRP for many patients? So, and in some countries, statin prescription is given only when they have an elevated total cholesterol level has gone, LDL level has gone, but they prescribe statin when the inflammatory markers are high. So things are evolving, things are getting different. So it's a inflammatory conditions. You just remember the slide, and in the last you can see increased steatosis and large adipocytes, inflammation, and insulin resistance. And we all say that, okay, this is non-vegetarian. Non-vegetarian is not good. So it contains phosphatidylcholine and those things which are present in the non-vegetarian food, red meat. And what happens is that phosphatidylcholine is converted into trimethylamine in the gut, and in the liver it's converted to trimethylene N oxide, TMAO. So TMAO production has a direct, the blood TMAO level and the risk of plaque and cardiac events, there is a correlation. So what really produces the TMAO is gut flora. So whether you can label with the evidence that the non-vegetarian is bad, no, but there is a causal correlation between TMAO and the risk of So inflammation helps drive the pathogenesis of atherosclerosis, and it has long been postulated that microbes are involved in the atherosclerotic process. And various autoimmune diseases like Hashimoto's thyroiditis, colitis, psoriasis, lupus, multiple sclerosis, and we are aware of the fact that a lot of people now develop autoimmune illness, and at least the serological examination, what we do, will show some positivity in most of the patients. And it is increasingly thought that the environmental triggers, including the microbiome, are partially responsible for the development of autoimmune illness. The infectious diseases has drastically reduced till 2000s, and now in the last 2 decades, we see the rise of autoimmunity, atopic dermatitis, allergic illnesses. The gut microbiota can influence or interfere with the immune sensing in the discriminating between self and non-self. I'll read the sentence once more. The gut microbiota can influence or interfere with immune sensing in discriminating between self and non-self. So, with this, there is a strong relationship for developing atopy, then atopic dermatitis, a lot of other inflammatory conditions, even bronchial asthma. There is role, and there is also evidences to say that the lung has its own microbiome. The skin, the atopic dermatitis, is actually the largest organ is skin. The skin has its own microbiome, and some decrease in variation of the bacteria on the skin is something is actually responsible for atopic dermatitis. And so what we can do, it's a practical tip to improve our gut microbial diversity. And see, it's what in my limited knowledge, what I can say is, see, it's not possible in a day like an emergency physician to come and do this thing. No, it takes years probably, or months probably, to make a little bit of difference in your gut. And there are different food items. Fasting has said to increase the gut microbial activity. When we give rest to our gut, the recycling phase of various microorganisms increases and the diversity increases. So giving a rest to your gut means good fellows start to multiply and repopulate. And exercise has said to do a good benefit, like sleep reduction, hygiene, and stress reduction. That's why more and more evidences now favor meditation, yoga, and in the sympathetic parasympathetic balance, all help to rejuvenate or to help in improving the gut microbial diversity. And since our processed food has no fibers, avoiding processed food helps. Also the processed oil. And we have to have diet which is rich in fiber. Like inulin. I don't know what exactly is inulin to prescribe to a patient. But inulin helps, that's what I learned. And include fermented food products, not the factory made yogurt, but we make it in home, probably can help, but not the factory made items. And avoiding unnecessary antibiotics, that is very important, because we doctors are forced to write prescriptions of antibiotics even for viral illness. So I'll not continue further. I think it's 9:45. So these are the practical aspects of improving your gut microbial diversity. And so in conclusion, so the complexity of fecal microbiota is actively being defined, and recent studies have shown that pathogenesis of many diseases result from microbiota-related dysregulation. Manipulation of gut flora may be an integral part of weight loss programs, and FMT reestablishes a balanced intestinal microbiota and results in the impressive cure rates in patients with recurrent CDI, that is a pseudomembranous colitis. And standardization of various protocols and randomized controlled trials are needed, and we are not pretty sure about a lot of aspects, but it's promising, and probably more science will evolve, and there are situations where you do a sample of your stool, saliva, blood. these three, and they study the complete microbiome. In that, they can predict how long you're going to live, and they can predict the future risk of your diabetes, and they can predict the progression. I mean, when you're going to become a diabetic, and even the glycemic target, the glycemic how it will be, like how ups and down, without checking the blood glucose level. So in one side we are moving in that direction, and I have not mentioned about neoplasms, cancer. The cancer treatment now has good. So we learned that some cancers respond to antibiotic. We had about some even multiple sclerosis responds with minicycline. No, I'm just saying. So see, there is a role of further, I mean, studies on this to give clarity, and we have to set up or think about a long-term goal to make a change. And it's not an emergency decision, but it's a long-term process that we all have to attempt to do. Thank you very much, and thanks for the opportunity.